Gibberellins are plant hormones that regulate growth and various developmental processes in plants. In particular, they are responsible for elongation growth of the stem that determines the height of the plant. Besides this, they also influence germination of seeds, flowering and development of fruits in plants. Gibberellins were first discovered in Japan by studying a fungal plant disease of rice. Chemically, they are terpenoid compounds, and over 120 types of gibberellins have been identified till now in plants, fungi and bacteria. Gibberellins enhance the activity of certain enzymes by inducing their synthesis in the seeds. One of these enzymes is alpha amylase that converts starch to glucose. In the alcohol industry, which uses starch as the starting material, gibberellins are used to enhance the activity of amylase for the conversion of starch to glucose. In agriculture, gibberellins are used to increase the height of the plants like sugarcane to increase the yield. They are also used to increase the size and yield of fruits like grapes and tomato, and to produce seedless fruits. Let us see in detail about gibberellins. A Japanese scientist, Aishi Kurosawa, discovered gibberellins in 1926 by studying a disease in rice that was caused by infection from the fungus Gibberella fujikuri. The name of this plant hormone is derived from the name of this fungus. The rice plants infected by this fungus grew abnormally tall, reaching almost 50% more height compared to the healthy plants. Although tall, these plants were thin, pale and sterile and produced no seeds. In Japanese, this disease was known as bekane, literally meaning foolish seedling. Foolish because it grew fast and tall, but produced no seeds. Later in 1935, two other Japanese scientists, Yabuta and Sumuki, isolated a chemical from this fungus which caused an increase in the height when applied to a healthy plant. They named this chemical gibberellin. The same chemical was subsequently isolated from plant sources and was found to be present in all types of plants, working as a plant hormone for elongation growth. The chemical is now known to be synthesized by fungi, plants and bacteria. Till now, about 126 types of natural gibberellins have been identified from these sources. The gibberellins are named as GA1, GA2, GA3 and so on in the order of their discovery. Gibberellic acid or GA3 is the most widespread and active gibberellin and was also the first to be structurally characterized. It is a tetracyclic diterpene acid having 19 carbons. All the gibberellins have 19 or 20 carbons with tetracyclic ring structure called the gibbane ring, but vary in their side chain attachments. The most bioactive gibberellins are GA1, GA3, GA4 and GA7. In general, the most biologically active forms are dihydroxylated gibberellins, which possess hydroxyl groups on both carbon 3 and carbon 13. Let us see the elongation growth promoted by gibberellins. The promotion of elongation growth by GA can be seen at the seedling stage using germinated seeds of dicotyledons. When the germinated seeds of amaranthus, radish or cucumber are grown in light in presence of GA, the growth of the hypocotyle, the stem portion below the cotyledons, is enhanced by GA. The enhancement in the growth of the hypocotyle shows a concentration response to GA in the range of concentrations from 0.1 to 10 microgram per ml. The growth of the hypocotyle is different when the seedlings are grown in complete darkness or in presence of light. There are three major differences in the seedlings: grown in darkness or in presence of light. In presence of light, the hypocotyle is shorter, cotyledons are more expanded, and the seedlings synthesize pigments like chlorophyll and beta-cyanin or anthocyanin. Treatment of light grown seedlings with GA not only promotes the growth of the hypocotyle, but also inhibits the synthesis of anthocyanin or beta-cyanin. This inhibition of the synthesis of pigments also shows a concentration response. Is there a relationship between the promotion of hypocotyle growth by GA and inhibition of beta-cyanin or anthocyanin synthesis? Beta-cyanin and anthocyanin pigments are synthesized from the amino acid precursors tyrosine and phenylalanine. These amino acids are also utilized in the synthesis of proteins. Application of GA enhances protein synthesis along with the growth of the hypocotyle and diverts the pigment precursor, inhibiting the synthesis of the pigments. Promotion of hypocotyle growth by GA is seen in light grown seedlings, but not in dark grown seedlings. Growth of the hypocotyle in darkness, which is higher than the growth in light, is also called etiolation growth. Since the growth of the hypocotyle in light in presence of external GA is also higher, we can ask the question whether the dark growth is dominated by internal or endogenous concentration of GA. Using a chemical inhibitor like chlorocholine chloride (CCC), which inhibits the synthesis of GA in plants, the growth of the hypocotyle in darkness can be inhibited. This inhibition of hypocotyle growth by CCC can be reversed by applying external GA along with the inhibitor. Application of GA to intact plants results in the elongation of the stem and increase in the height of the plant. This increase is due to elongation of the internodes. Measurement of internodal length in the control and the GA treated plants will show the difference in the length of the internodes. Some plants like cabbage are naturally dwarfed. Application of GA on these dwarf plants causes bolting, that is elongation of the stem and spreading of the leaves. Plants like pea and maize become dwarf by genetic changes or mutations. The dwarf mutants lose the capacity to synthesize GA. Application of GA removes dwarfism in these mutants by inducing internodal growth. Mendel's dwarf peas were genetic mutants of this kind. height of the pea plant was one of the several genetic traits Mendel used in his experiments. GA has a major role to play in seed germination. After sowing the seed in the soil, germination is the first phase of development of the seed. The seed absorbs water from the soil, sprouts, starts growing, and the seedling comes out of the soil. The growth phase from the time the seed is sown and till the time the seedling comes out is supported by the reserved food material present in the endosperm of the seed. The seeds of cereals like rice, wheat and maize have starch as the reserved food in their endosperm. Let us take the example of wheat seed. We can distinguish two parts in this seed, the upper part containing the embryo and the lower part made up of only endosperm. The root and the shoot start growing from the embryo in the upper part when the seed germinates. We can cut the seed into two halves, separating the embryo half from the lower half. When these two halves are separately incubated on an agar plate containing starch, we can observe that the embryo containing half digests more starch than the other. The amount of starch degraded is observed on the plate after 24 hours of incubation by staining the undigested starch with iodine potassium iodide solution. The clear circular zones around the half seeds indicate the digestion of starch. Starch is a polysaccharide of glucose and is digested by the enzyme alpha amylase. This enzyme degrades starch by breaking the glycosidic bonds between the glucose molecules randomly to form oligosaccharides, disaccharides, that is maltose, and glucose. The embryo part of the seed degrades more starch because of more production of amylase enzyme. This is due to the gibberellic acid produced and released by the embryo. In the seed, outside the endosperm, there is a layer of cells called aleurone layer. The cells in this layer are the seat of amylase synthesis and secretion. GA promotes the synthesis of amylase in these cells. This promotion is brought about by overexpression of genes responsible for producing the specific messenger RNA for the enzyme, which in turn are translated to amylase. Hydrolysis of starch is necessary for the germination of starchy seeds and conversion of starch to glucose to support germination and seedling growth. Secretion of GA by the embryo initiates this process in the seeds of cereals rich in starch. All the seeds are not rich in starch. The seeds of pulses are rich in proteins and the oil seeds are rich in lipids. Besides amylase, GA also promotes the synthesis of other hydrolytic enzymes like proteases and lipases. These enzymes are responsible for the degradation of proteins and lipids. In general, irrespective of the type of reserved food present in the seeds, GA helps in the enzymatic degradation or hydrolysis of the reserved food to initiate germination. Once the seedling comes out of the soil and forms cotyledons or leaves, the growth of the seedling will be supported by photosynthesis. All seeds do not germinate soon after their production. Instead, some of them may get into a period of dormancy. The dormant seeds do not germinate even when they are provided with favorable conditions for germination like moisture and temperature. One of the reasons for seed dormancy is the presence of certain natural compounds which inhibit germination by acting as GA antagonists. The plant hormone abscisic acid, the phenolic compound coumarins are two examples of natural compounds which can inhibit germination. Seeds will be able to germinate only when these compounds are degraded naturally over a period of time. Similarly, there are dormant buds, especially in the plants of temperate regions, which suppress growth of shoots by accumulating inhibitors like abscisic acid. Dormancy of seeds helps in the dispersal of seeds. The time period required for degradation of natural inhibitors allows the seed to gain distance away from the parent plant before it can germinate and produce a new seedling. The plant can spread to a large area in this manner and reduce competition for water and nutrients. In case of dormant buds, the plant avoids forming new shoots in adverse weather conditions like winter period in temperate zones. Once the spring arrives, the buds are ready to form new shoots. In both these cases, the natural dormancy can be broken by the application of external GA. The external GA changes the concentration ratio of inhibitors to promoters inside the seed or a bud and allows germination or shoot growth. Let us see the effect of GA on flowering. Depending on the day, night or light dark cycles required for flowering, the higher plants are broadly divided into three categories. These are short day plants, long day plants and day neutral plants. The short day plants can flower only when the light period is shorter than a critical length of daylight. The long day plants are those which require longer than a critical length of daylight. The day neutral plants are insensitive to daylight and can flower in either condition. If a long day plant is grown in non-inductive condition, that is by providing less than critical day length, then they do not flower. For example, if a long day plant has 10-hour light period as critical day length required, then growing them in a daylight of less than 10 hours will not induce flowering in them. Under such a condition, application of GA induces flowering in them. In other words, GA can fulfill the requirement of critical day length in a long day plant even under non-inductive conditions. Some plants require exposure to low temperature before flowering. This process is called vernalization. Application of GA can overcome this requirement in some of the plants. Let us see the effect of GA on fruit development. GA can induce parthenocarpic development of fruits, that is development of fruit from a flower without fertilization. Such fruits will be seedless. GA can induce parthenocarpy in apple and pear. There is a seedless variety of grapes called Thompson seedless grapes. This is a pale green, seedless, oval shaped variety of grapes originally from Ottoman Empire. William Thompson, an early grower of grapes from California, is credited with introducing this variety to USA, and this variety came to be known as Thompson seedless grapes. The dried grapes produced from the green grapes are called raisin. Application of GA increases the number and size of the grapes by elongating the fruit stalks. GA application also enhances the size of the tomato fruits. In citrus fruits, application of GA delays ripening and increases the shelf life of the fruits that help in transportation. We can compare the action of auxin and gibberellins. Although both auxins and gibberellins promote cell elongation, longitudinal growth and parthenocarpy, they also differ in many ways. Polar transport, root initiation and prevention of leaf abscission are associated with auxins, but not with gibberellins. Similarly, breaking of dormancy of seeds and buds and elongation of dwarf plants are the responses induced by gibberellins, but not by auxins. Gibberellin signal transduction. Although many genes stimulated by gibberellins in regulating various biochemical and physiological processes have been identified, the mechanism of signal transduction has been extensively studied in relation to the synthesis and secretion of alpha amylase. This enzyme is secreted by the cells of aleurone layer which surrounds the endosperm of cereal grains. A model has been proposed for understanding the mechanism of stimulation of genes by GA by studying the events in these cells. GA first binds to a receptor present on the plasma membrane of the cell. The GA receptor complex then reacts with a heteromeric G protein of the membrane. G proteins can activate the enzyme guanyl cyclase, which synthesizes cyclic guanine monophosphate from GTP or guanine triphosphate. Cyclic GMP can regulate ion channels, calcium levels, protein kinase activity and gene transcription. In aleurone layer cells, two separate signal transduction chains are initiated by cyclic GMP, a calcium dependent channel and a calcium independent channel. A signaling intermediate is activated through the calcium independent pathway, which binds to a repressor protein in the nucleus. This repressor, known as DELA repressor proteins, is inactivated and degraded when it is bound to GA signal intermediate. This inactivation allows the expression of MYB gene. MYB genes produce transcription factors which are known to regulate growth and development in plants. The MYB genes code for GA MYB messenger RNA, which is translated to GA MYB protein outside the nucleus. This protein acts as the transcription factor, enters the nucleus and binds to the promoter gene of alpha amylase. The alpha amylase messenger RNA transcription is activated and later translated to alpha amylase on the rough endoplasmic reticulum. The alpha amylase synthesized and enclosed in secretory vesicles are released from the Golgi bodies. The secretion of alpha amylase requires calcium calmodulin dependent signal activated by GA. Let us see the applications of GA. GA is widely used for increasing the stalk length of seedless grapes. The seedless grape bunches are compact and the growth of the berries is restricted because of short stalks. GA treatment stimulates stalks to grow longer and removes the compactness. The treatment also promotes elongation of the fruit. GA is also used to promote the growth of apple and pear. Application of GA delays fruit ripening in citrus fruits and helps to extend their shelf life. Sugarcane crop stores sucrose in the stem and can grow tall. By the application of GA, the length of the internodes of the stem can be increased. Since sucrose is stored in the stem part, the yield of the crop will also increase. It is estimated that application of GA can increase the yield of raw sugar cane up to 20 tons per acre, and the resulting sugar yield per acre will be 2 tons. In the brewing industry, malting is the first step in which barley seeds are allowed to germinate at temperatures that maximize the production of hydrolytic enzymes. GA is used to speed up the malting process and to enhance the production of amylase enzyme. The germinated seeds are pulverized to produce malt, a mixture of starch degrading enzymes and partly digested starch. The malt is used to convert starch to glucose, and later glucose is fermented to ethanol by yeast. GA is used in the breeding programs to reduce the time of seed production in conifer trees. GA reduces the long juvenile period of conifer trees. GA also promotes the production of male flowers in cucurbits and stimulates bolting in rosette crops like beetroot and cabbage. These effects of GA are used in the commercial production of seeds in these plants. Inhibitors of GA like paclobutrazol and ansimidol are used to reduce height of flowering plants like chrysanthemum and lilies. Another application of the inhibitors is in restricting the growth of roadside shrub plantings. Inhibitors are also used to prevent lodging in certain crops by shortening the internodes. Lodging is the bending of stems to the ground caused by the weight of water collecting on the ripened heads. Shortening the internodes reduces this tendency and increases the yield of crops. In conclusion, gibberellins are an important class of plant hormones regulating several growth and developmental processes in plants. They have a major role in the germination of seeds, in the control of plant height, and in the regulation of flowering. Gibberellins are commercially important in the brewing industry, in increasing the yield of sugar cane, and in the production of Thompson seedless grapes.